Nucleotide excision repair is really important because unlike something like base excision repair or mismatch repair, where it just changes one nucleotide to fix something or a small portion, nucleotide excision repair fixes a large portion of things, which can be really effective for preventing or repairing a lot of large and really dangerous types of damage. So the way it works, basically, there's a couple of different ways depending on if it's prokaryotic or eukaryotic, bacterial or eukaryotic. In bacteria, you have a couple of proteins, enzymes that come through, two in particular, UVRA comes in and marks damage, finds damage. UVRB comes in and is able to unravel that part of the DNA, the damaged part of the DNA, and has that marked. And then an enzyme called UVRC comes into that marked place and cuts it on either end. So basically UVRA and B are here. B unravels it. C comes in, cuts it on either end. UVRD pulls that out, and then it's able to resynthesize and put it back in, and ligase seals up the ends. So that was pretty simple. Eukaryotic is very similar. It just has a couple of different enzymes names. There's also two different types in eukaryotic. There's global and then transcription coupled. Global is basically an enzyme, XPC, which is floating around and it just finds things wherever, issues wherever. But because eukaryotes have so little portions of our DNA that are actually protein coding, we have one called transcription coupled repair, which is basically where an RNA polymerase 2, is going down transcribing all of the DNA that is protein coding, and if it runs into an issue, whether that's backbone break, double strand break, any sorts of things like that, large portions that are damaged, then it stops and it basically unloads markers. It's basically carrying them around in like a backpack. And so this is XPB and XPD that it unloads. They're very similar to UVRA and UVRB in prokaryotes, where they mark it, unwind it, and then XPF, XPG come in and they cut it on either end, just like UVRC does in prokaryotes, and then that's able to be brought out and resynthesized, put back in. And then again, ligase seals it up. So this is really important according to many studies, particularly a study by the the National Library of Medicine, particularly in relation to Xeroderma pigmentosum, which is basically caused by a defect in nucleic excision repair. Because nucleic excision repair doesn't work when someone has this, it's not able to find those larger portions that are damaged, and it's not able to replace them, and it causes a lot of cancer because it's not able to fix the things that are broken, right? And so a lot of times people with this Xeroderma pigmentosum, they get skin cancer a lot because the UV damage that comes from the sun when they go out there, it happens to all of us all the time. We get the UV damage, and if it weren't fixed, it's very likely that it turns into cancer. But most people have this ability to repair it with nucleic excision repair. Those who don't have it. and they aren't able to.